Bismillahirrahmanirrahim. Welcome back, friends. Welcome to another video session from Chemi Biology. In the previous video, we talked the membrane fusion inhibitor and the drugs and their mechanism of action. Now this is the third video regarding 20 viral drugs, and in this video lecture we will talk about the protein uncoating inhibitor. Find the drugs which are used in protein uncoating inhibitor and their mechanism of action and the resistance which are activated against these drugs. Fine. So now let me write. The drugs which are involved in protein uncoating inhibitor, we usually call that Amantadine and second name is Rimantadine. So these are the names of the drugs which are used in protein uncoating inhibitor. Fine. Now if we talk the broad spectrum of these drugs, so what does broad spectrum means? Broad spectrum means on which kind of viruses these drugs are activated and these drugs show best result. So the broad spectrum of these drugs are that these drugs are activated against Parkinson's disease. And also activated against influenza virus. So against the Parkinson's disease and influenza viruses, these drugs show best result. Fine. So this is the broad spectrum. Now let's talk in a detail the mode of action of these drugs. Now before going to explain the mode of action, you need to understand the structure of the structure of the viruses. Fine. Now to explain these drugs, now to explain the mode of action of these drugs, you need to understand the structure of the viruses. Fine. So let me draw the structure of viruses, that how the viruses will be look like in this case. In this case, let's suppose this is the envelope area. Fine. Inside this envelope area, they have capsid. So inside the envelope area, there will be a capsid, and inside the capsid, there will be a genome, any kind of the genetic material, DNA, RNA. Fine. So outside there are some protein, the glycoproteins arises. Sometimes these proteins are called so now these are the spikes, and these glycoprotein, or we can say spikes, are responsible to attach on the host membrane, on the host cell receptor, and then they will be engulfed. So, and this one area as I draw, these are the neuraminidase enzyme. Neuraminidase enzyme, and their function, everything, their activity will be talking the next video lecture. But here you just understand that this one area is actually responsible for the attachment to the host receptor, and then they will be internalized. Fine. Let's suppose the viruses. In this case, let's suppose this structure represents the influenza virus, but it is a rough form of the influenza virus. Fine. Let's suppose this is the structure of the influenza virus, and there are also some other proteins. Now this one area, as I draw, this one also the protein area, but we call that M1 protein. We call that, which one? We call that M1 protein. And there are also some other proteins. Let me draw these proteins with the green one, which have the special channels. Fine. So these proteins have some special channels. We call that proton selective channels. Due to these channels, the proton will be emitted out, and once the proton are released out, so they will put some pressure on the envelope area, or they will put some pressure on the endosomes, by which the protein, mean this capsid protein, will be degrading, or start degrading, or this capsid protein will uncoating, and the genome will be released out. So due to this, we call actually this protein, we call that M2 protein. What does M means? M means matrix. Fine. M means matrix. So matrix 1 and matrix 2. So matrix 2 are responsible for this purpose. Now, so let me draw the hydrogen ions here. So it releases the hydrogen, due to which the capsid area will start degrading. Fine. So this is the function of this M2 protein, and once the capsids start degrading, then their genetic material will released out. And once the genetic material is released out, let's suppose it is released out inside the host cell. Fine. So let's suppose this is the genetic material which are released out, and after that replication process will be started. Replication will be started. Replication start. Fine. So this is the function of the, so this is a little introduction to this virus structure. Now where the drugs actually, which side the drugs actually targeted. This amantadine and rimantadine actually targeted these M2 proteins. Fine. So if I draw the structure of, let's suppose this black area, let's suppose these black areas represents that, let's suppose this black area represents that the drugs are here, and the drugs will block these spores or these channels. Due to this blockage, the hydrogen ion, or we can say the proton, will be not released out. Fine. So these are the drugs. These are the drugs. So once the drugs will bind, what the drugs actually do, the drugs will bind to these pores or these channels, by which it can prevent the release out of the hydrogen ion. Or we can also say, sometimes if the hydrogen ion are not present inside the viruses, so they will take the hydrogen ion outside this cell environment. Fine. So they will not take the hydrogen ion from outside, as well as the hydrogen ion which are present inside the viruses will be not released out. So the genome of the viruses will remain inside, because due to this hydrogen ion, the protein are not uncoated, means this capsid area will not uncoated. So finally the replication process will be halted. Fine. So this is the mechanism of action of these drugs. Now if we talk the resistance, so the resistance occur against these drugs very quickly. How? Sometimes the hydrogen ions, it make pressure hydrogen, or we can say the protons. What the proton actually do, it make a pressure. So once the pressure, means when the proton, not protein, sorry, proton. So once the proton pressures are activated, so what happen? They will eliminate the drugs, means they will eliminate the drugs. So these drugs, let's suppose this is the drugs, so these drugs will be eliminated out. So where the drugs will be eliminated out. So now this is the drugs, and the drugs will be eliminated out due to high pressure on these proton, on these M2 protein, on this proton channel, we can say. So the drugs will be eliminated out. Fine. So this is means the resistance will be occur very quickly against these drugs. This is the first resistance. proton pressure. And the second resistance against these drugs, we say that changes in amino acid. Changes in amino acid. Fine. What does it mean? It means that you better know that protein is made up of amino acid. So let's suppose this M2 protein is made up of amino acid, and we call it valine, isoleucine, or leucine. Fine. Let's suppose these are made up of these 3 amino acid or 4 amino acid. We can get tryptophan. If the sequences, or if any one of the amino acid are substituted, so what happened? Let's suppose the amino acid which are involved in this case, let's suppose we call that isoleucine, valine, tryptophan. Fine. If any amino acid are changed, let's suppose this one amino acid is eliminated, or we can say this one amino acid is substituted by another amino acid, and we call that leucine. Then what happened due to these changes? The drugs will also not affect it. The drugs will come here and they will again eliminate it out. This is the second resistance. Fine. So this is the resistance which are activated against these drugs. Now if still we are giving these drugs to the patient, and although the virus show resistance, so then these drugs have some side effect, or we can say adverse effect. And these adverse effect, let me write here. So the adverse effect. So the adverse effect in this case are what? No, let me write the names. Insomnia. Insomnia means that the patient will not sleep at night. The patient sleeping will be disturbed. Second is skin rashes. Means there are some red spots appear on the skin. We call that skin rashes. Third one, we say that atherosclerosis. Fine. Means that the fatty acid will be deposited in the artery. So these are the side effects of these drugs. Fine. So that's fine. So that's all about the protein uncoating inhibitor. And in the next video, inshallah, we are going to talk about the nucleic acid inhibitor. Means the drugs which are involved in the nucleic acid inhibitor, the mechanism of action of these drugs, and the resistance which are activated against these drugs, and the adverse effect also of these drugs. Fine. So stay tuned and continue to watch. Thank you so much.